Thank you for coming to my talk. Today I'll be talking about HistomicsTK. It is a Python toolkit for analyzing histopathology data. Firstly, I would like to get a sense of my audience. So how many people here do image analysis or computer vision? Okay. How many people do machine learning? Okay. How many people are into biomedical? Great. Okay, that's good. So first, I'll start with telling about the people behind this work. HistomicsTK is being developed as part of collaboration between Kitware and Emory University. The funding is coming from an NIH grant from a program that aims to develop information technology tools for cancer research. So there are two faculty members at Emory University, Lee Cooper and David Gutman. And Kitware is the subcontractor on this grant. And these are all the wonderful people behind this work. It's not me alone. So first, I'll take a moment to tell you about Kitware. Kitware is an open source scientific computing company, and we have people with expertise in the domains that you see here, mainly medical image analysis, and computer vision, data and analytics, and high performance computing and visualization. We are heavily involved in developing a lot of open source toolkits. Some of the popular ones you probably might have heard are ITK. There was a talk on ITK by Matt a couple days ago in Pydata. And there is Slicer, which is a desktop application to view medical images and analyze them. JC here is the Slicer guy at Kitware, so if you have questions, you can go to him. And there's VTK, which is a very popular visualization library, and CMake, which is a cross-platform build system. And there are several other toolkits which we work on. So before I start with HistomicsTK, I would like to introduce you to histopathology if you haven't heard of it. So what is histopathology? It is about examining thin slices of diseased tissue that are fixed on glass slides. You put them under a microscope and look at them at cellular resolution. And you usually apply chemical stains to them. You can think of them as color dyes that basically highlight specific structures in these images, for example, nuclei, cytoplasm, or other kinds of structures or proteins that you're interested in, right? And histopathologists typically look at the tissue architecture, and each disease has a particular manifestation on the tissue architecture. They look at the morphology of the cells and nuclei and extracellular structures that gives them information about what's going on with the disease or whether the disease is there or not. Histopathology is pretty much the gold standard in the diagnosis, staging, and prognosis of several diseases, including most types of cancers. So recently, there were whole slide imaging systems developed. These basically scan the entire slide and produce huge 2D images. So in the center, you see an image. Basically, it's at the base resolution, it's at 40x magnification. It's about 50,000 pixels across, so 50,000 pixels tall. That's about 2.5 billion pixels. In uncompressed format, it's about 7.5 gigabytes of data, and each of these images typically contains about 1 million nuclei that you want to analyze, right? And there are about over 10 whole slide image system vendors right now, and if you look at the PubMed trends, there is an increased interest in whole slide imaging currently. And one of the biggest public data resource for this is the Cancer Genome Atlas that is spearheaded by the National Institute of Health. This contains data for 33 different types of cancers from 11,000 patients, and it contains data, 7 different kinds of data, and that's all in all about 2.5 petabytes of data, and there are lots of studies that are going on based on this data. So a few years ago, before we started on the development of HistomicsTK, these two members that you see on the right from Emory University developed what is called a cancer digital slide archive. It's basically a viewer to look at all the whole slide images that are part of the Cancer Genome Atlas data set. So they ingested about 32,000 whole slide images that come from about 19,000 patients and 33 different cancer types. That is about 15 terabytes of data. And there is link of each whole slide image to other data modalities as well. So that's a humongous amount of data. And considering the scale of these images, you can imagine that it takes a lot of time for histopathologists to basically go through the image and arrive at a diagnosis or a prognosis. Right? So obviously there has been interest in developing computational techniques to inject some objectivity into this process. Each disease has a unique manifestation on the tissue characteristics of these images. They alter the nuclear morphology in a specific way. So what computational histopathology involves basically developing image analysis and machine learning algorithms to basically quantify these characteristics, these tissue phenomena with some quantitative features. And then there are two goals. One goal is to take these imaging biomarkers or features extracted from these images, and you correlate them with clinical survival data or clinical event data to build survival models to tell how aggressive the disease is or how long the patient will survive. And with that, you can build tools, decision support tools to recommend what kind of treatment the patient should go through. That is one goal. And the second goal is basically to understand tumor biology itself. You basically correlate these imaging biomarkers with other genetic variables to understand the correlations, and hopefully that will one day, that will probably lead us to developing better cancer drugs. So I've listed out the key requirements to make all this research possible. First thing is you need a web-based platform for data management and also analytics. The data sizes are too high for moving the data around, so you have to bring the analysis to the data. And second, there's a need for an open source platform as well. There are not many open source tools in the pathology domain as compared to radiology or genomics. And we want it to be open source, but also with a license that permits commercialization. And there's a whole lot of data management needs that come with it, and we need ability to visualize these whole slide images at different scales to support the pathologist's workflow, and we need ways to do markup on these images, do annotations on these images, and we need a toolkit. So the algorithm community needs a toolkit with state-of-the-art algorithms to build upon. And they also need a platform to disseminate their new solutions, new analytic solutions somewhere for wider use with the community. The pathologists also need a platform to evaluate and pick among all the state-of-the-art algorithms that are there. And lastly, and most importantly, we have to build for the community and engage them. That way we'll collectively meet these scientific goals faster. So our solutions are twofold. We have one web application called Digital Slide Archive. It aims to fulfill the data management needs of whole slide images. And the other is HistomicsTK, which aims to fulfill the analytics needs of this data. And both these tools are open source with an Apache 2.0 license. And both these tools are based on what is called a Girder, that is under active development at Kitware. What is Girder? Girder is a Python framework. It started as a data management platform, but now it's more than that. It is a Python framework that helps you build web applications that store, manage, and process scientific data. That's the way I would like to put it. So it has RESTful access to all the data that is stored inside, and it supports multiple backends. You can have the data on the file system or Amazon S3, HDFS, or wherever. And then there's search capabilities, which can be improved. And you can have users, groups, and there is a Girder plugin which lets you do distributed task execution that is based on Celery underneath. And there's a very nice plugin infrastructure that helps you build server-side analytics applications. I'll do a demo of this in a bit, and if you want to get a feel of it, you can go to data.kitware.com. It's based on Girder, so you get a feel of it. I'll start with the digital slide archive. This is basically an extension of Girder. These are some screenshots that I put if the demo doesn't work. So let me try. So this is the dashboard of the digital slide archive. It's basically an extension of Girder with a few features that are specific to histopathology. So there are collections which are essentially sort of folders. It's going to feel like a file explorer to you. So we have ingestion scripts that ingest the data from the cancer genome atlas, and these are all different cancer types. And if I go into one of the cancer types, in a bit, yeah, these are all the patients for each cancer type. There are people with the disease, and there are also people without the disease, so you can do machine learning on them. Yeah, it's a bit slow, but okay, I have a backup. Yeah, it's supposed to show thumbnails of those images, and if I go into one of these images, there's an embedded viewer, so you can link custom metadata tags to each of these images, like clinical information, stain type, or any other information that is there. Right? And so there is, let me go to the other one. So there are different kinds of asset stores. You can have the data on file system or MongoDB, Amazon S3, or HDFS. You have user and group management. And most importantly, there is all the data is there are REST endpoints to access the data. If there's an extensive amount of RESTful access, for example, I can download the file, upload the file through REST endpoints. Okay, it's a bit slow. Yeah, so those are all the REST endpoints, and hopefully my viewer loaded. Okay, it's not loading, but anyway. So it's supposed to have an embedded image viewer, so you can zoom through these images and pan and look at them. Right? And let me go further. So coming to HistomicsTK, HistomicsTK is being developed with three kinds of communities in mind, the algorithm research community and the pathologists and cancer biologists. And there are two modes of using HistomicsTK. One is it can be used as a pure Python toolkit for algorithm development. It aims to provide state-of-the-art algorithms for each component of the data analysis pipeline. So if you're working on one component of the pipeline, you can use algorithms from HistomicsTK for the rest and develop on just that one piece. And it can also be used as a Girder plugin for web-based analysis. In this mode, it provides a framework to easily expose analysis modules, command line analysis modules on the web. It provides a platform for researchers to disseminate their solutions easily for wider use with minimal effort on their part. I'll do a demo on that. And it provides also a platform for pathologists and biologists to look at what analytic solutions are there from algorithm labs and pick and choose from them. And it has a backend engine for distributed task execution based on Celery. Coming to the first mode of use as a Python toolkit, these are all the algorithms that we are trying to build into HistomicsTK. Beginning with pre-processing, there are already some functions to do color normalization. If there is non-uniform staining in these images, you can correct for them and normalize them. There's color deconvolution, which involves basically you have an image wherein multiple stains are applied, and you algorithmically separate these stains. Basically, if you have a stain that highlights nuclei and a stain that highlight cytoplasm and extracellular structures, you can separate them and then run nuclei detection algorithms on them. We already have some nuclear morphometry algorithms there. We are working on cell and nuclear classification algorithms and immunohistochemistry quantification, which is about staining them with proteins if you're looking for certain proteins that you're interested in, and tissue or region classification, and lastly integrative analysis with genomic data. And we use read the docs for our documentation. That is, this is the API reference that shows a high-level view of the kind of functions that are there in HistomicsTK. And coming to the second mode of use as a Girder plugin for web-based analysis, this is the dashboard of web-based analysis. That's a screenshot just in case the demo doesn't work. Yeah. So there is a way to open the image in the linked Girder instance, and then there is analysis. Maybe I will explain this first and come back to this. At the core of this is basically a very simple framework to expose command line analysis modules, currently written in Python or C++, but we can add other languages as well. This framework exposes any command line analysis modules on the web. There's a protocol that you need to follow. So what the developers need to do is they create a folder in their GitHub repository for each analysis module. They write an XML file which describes the inputs, the types of the inputs, the parameters, any default values, and outputs of the output of the algorithm. And then they write their code, which basically adheres to this XML spec. So let me quickly show you on command line how this works. So, oops. Is it big enough? Bigger? Okay. Better? So in HistomicsTK itself, there are 4 analysis modules. I'll go into one of them. And you have a folder for each and every analysis module. Inside there's an XML file which describes the input parameters and outputs, and there's a Python file which actually does the analysis. And so this is how the XML spec looks like. This is based on the slicer execution model. This is what the slicer, the desktop application that I was talking about for medical imaging uses. And we made a way to expose these modules over the web. And it was very successful with Slicer. There are hundreds of plugins written by the community. So as you can see, there are like input. There's an input parameter, and there are some stain color parameters, and these are the outputs that this module produces. And then there's the actual Python code that does the analysis. And what we have is a Python package called CTK CLI, which does the command line argument parsing and help for you. Let me show you what this does. If I do --help, so you basically get usage help on how to use this command line module. What it's basically doing is it's parsing the XML file and generating this for you. And it also has --XML endpoint, which basically throws out the whole XML file. That is what is used to automatically generate a REST endpoint to use that module over the web. So let me go back to my demo. The other thing that you write is basically a JSON file which lists all the analysis modules that you want to expose. Hopefully this comes up. Yeah, okay. And then you write this JSON file, and then you write a simple Docker file. You can derive from the HistomicsTK's Docker image, which will install HistomicsTK and all the dependent libraries for you. And then you install any requirements of your analysis module, and then you call an entry point that we provide. And let me show you what that does on the command line again. Actually, this bigger. Okay. How am I doing on time? Okay. So I have a few Docker images that were generated that way. And let me run the HistomicsTK's Docker image. If I do --help, so it basically generates a help on how to use the Docker image. You can basically run any CLI command line module inside the Docker image. And then there is a --list CLI endpoint, which basically throws out that JSON file contents. So there is a piece of code which basically analyzes the JSON file and then goes to the folder which has the analysis module, picks out the XML, and then generates the REST endpoint automatically for you. So, and let me go back here. And so you basically build the Docker file. There is a GitHub hook to automatically build the Docker image and upload it to Docker Hub. And there are endpoints wherein I can simply go and add. You can send me an email saying, hey, I want to host my Docker image, which contains the algorithms developed in our lab on your website. And I can simply go to a REST endpoint and add the Docker image. And it shows up here. And for each Docker image, there could be multiple versions. And then for each Docker image, there are a bunch of analysis modules that it exposes. And the one you see on the left is basically the widget tree to set the parameters, inputs and outputs for the analysis module. And all this is also automatically generated from that XML file the developer writes. So there's like very minimal effort on the developer side to expose it on the web. And yeah, so we also have the capability to render annotations like boxes, circles, contours, all that kind of stuff. All right. And what this does is it gives a nice platform that pairs different kinds of communities. The pathologists and biologists have data questions. The algorithm researchers get the analytics needs from that community, develop their algorithms. They can use Docker. They can use our mechanism to push it to an image on Docker Hub, and then we can host it on the website from where the pathologists and biologists can access all the analytic solutions, compare between them, and choose between them. Right? And lastly, some of software process stuff. HistomicsTK is on GitHub. We have documentation on Read the Docs. We are working on more documentation. There's quite a bit there, but we are working on more. We haven't made our official release yet. We are waiting on improving the code coverage a bit. We are about 60% code coverage. We want to improve it a bit more. We use Sphinx to automatically generate API documentation from the doc strings inside the functions. And there are some usage examples using Jupyter notebooks on Read the Docs. And we use Travis CI for continuous integration. And we use Vagrant and Ansible to ease the installation process. You can go to Read the Docs for more details. And these are all the future work that we are planning. We'll probably make a release sometime soon. We'll be adding more algorithms. We are looking into ways to speed up algorithms using Python and using Python wrapped ITK for whatever we can. And we are working, so in the next year, we'll probably take up big data processing challenges. Basically, we want to process the we want to parallelize the processing of individual tiles of these whole slate images. So these are basically stored in a pyramidal fashion, and at each level of the pyramid, the image is broken down into blocks or tiles. Right? We want to parallelize the processing of these blocks. We are looking at technologies of how to do this. We'll be venturing into the omics side of things, integrate do integrative analysis with genomic and clinical data, and a bunch of other stuff. And Kitware is hiring. These are all the positions that are currently up on jobs.kitware.com. If you find one of them interesting, go and apply for them. Thank you. So.
Thank you. We have time for questions. When you ask a question, please take the microphone so we can get it on the video.
Do you find difficulty debugging in the whole web workflow?
Oh, a lot.
Algorithm problems?
A lot, yeah.
What are your experiences and suggestions?
So we had very minimal exception handling before. We increased that a lot. Girder has a way to log exceptions wherever they occur. So right now everything goes into a log file on the server. You can go and look there. So this mix of web application and scientific development, there are like some weird debugging issues that you face with it. So yeah, that's there. So we are trying to make it as easy as possible for the algorithm developers. We want to make it feel like they're just developing a command line analysis module. They just have to import a particular Python package to do command line argument parsing. And it's basically an executable. They can test it on local files on the command line. And as long as they use the CTK CLI package, we'll be able to easily expose it over the web. So yeah, we want to make it as easy as possible for the algorithm developers. They shouldn't have to deal with complexities of exposing their algorithms over the web. And likewise, the pathologists and biologists, they don't want to develop algorithms. They just want to look at different state-of-the-art solutions that are out there for each analysis component, and then compare them in a black boxes fashion, and then choose among them based on what works on their data. Yeah. So.
Next question. Well, I have a quick question. Are your primary users histopathologists, and how do they feel about their new workflow?
We haven't brought this to them yet. We want to do that after our first release. Our collaborators at Emory University work very closely with pathology labs in the university. So they'll be our first users, and then we'll get a feel of how things are working. So,
thank you.
Yeah.
All right. Thank you very much.
The viewer now works, so you can just zoom through the images and see them. Yeah, the curse of the live demo.
All right. Thanks, everyone.